Okay, the learning objectives for this video is to explain the importance of the Anfinsen experiment, describe characteristics of the primary structure of proteins that limit folding, describe the primary interactions that are necessary for second, third, and the fourth levels of protein structure. I want you to be able to differentiate between alpha helices and beta pleated sheets. And finally, I want you to be able to explain the difference between protein motifs and domains. So the Anfinsen experiment occurred in the 1950s, and what these researchers did was to take an enzyme that was in its native state, in its functional conformation, and they could show that it was catalytically active. It was an enzyme and it was capable of catalyzing its reaction. They then added several denaturants to the solution in which this protein was dissolved, and they denatured it. They broke disulfide bonds, they broke all of the various weak interactions, and they showed at this point, because it was unfolded, because it was denatured, it no longer had any function. And then they removed all of the denaturants. They removed the mercaptan ethanol, they removed the urea, and without anything else, we're talking about a protein dissolved in an aqueous solution. There's no other proteins there, there's no other macromolecules around, it's all by itself. And they showed that after removing the denaturants, it went back into its native state and was able to be catalytically active again. And so I've got this question here: what is the significance of this experiment? And what it demonstrates is that the information necessary for a protein to fold into its functional conformation, the conformation that allows it to do what it is supposed to do, all that information is right here. The primary structure of that protein, the sequence of amino acids connected together by peptide bonds, is all the information necessary for that protein to fold into its native conformation, its conformation that leads to the functional state. So here we have the 4 levels of protein structure: the primary, secondary, tertiary, and quaternary. Now the primary structure, as I said, is what determines all of the rest of this, and indirectly the protein function. Okay, so the peptide bond that connects amino acids together is unusual in that, because of resonance, although it looks like a single bond, it actually acts more like a double bond, which means there's no rotation around those peptide bonds. In addition, the bonds that do rotate in the polypeptide chain backbone, the phi and the psi bonds, they can't rotate 360 degrees. There are certain angles that are impossible because of steric hindrance. So there are some limitations to how this chain is going to fold, simply because of the resonance of the peptide bond, the fact that it acts as a double bond, and because of steric hindrance. Okay, be sure to watch that video about the Ramachandran principle. Okay, the secondary structure of proteins is dependent upon hydrogen bonding within the backbone of that polypeptide chain. What do I mean by backbone? What I mean here is, what we have is, here's an amino acid, here's an amino acid, here's an amino acid, and they're all connected by peptide bonds to peptide bond, peptide bond, peptide bond. So we have this kind of this backbone, NCC, NCC backbone, and this is the alpha carbon to which the R groups are bound, here, here, and here. And the secondary structure doesn't include the R groups. What it includes is hydrogen bonding between this hydrogen here of the peptide near the peptide bond and this double bonded oxygen here near the peptide bond. Now the two of them don't hydrogen bond together. Instead, they hydrogen bond with others amino acids farther down. So for example, if we took a look at the hydrogen bonding pattern for an alpha helix, a very common secondary structure, what we see, it's about 4 amino acids away. So this oxygen is hydrogen bonding with 1, 2, 3, 4, the hydrogen here, 4 amino acids away. And there's several different ways a polypeptide chain can fold in order to accommodate all of this potential hydrogen bonding. The alpha helix is this kind of helical folding where it's making this winding pattern here around an axis. And in so doing, it forms a hydrogen bond with the amino acid 4 amino acids away. This is a very, very common form of secondary structure. There are things, however, if you take a look at the sequence of amino acids within a protein, there are ways you can look and predict that would probably not make an alpha helix. And these are what I'm calling the alpha helix breakers. So if you see an abundance of these amino acids or an abundance of these situations within a sequence of amino acids, you're probably pretty sure that you're not going to have an alpha helix there. Branched amino acids. Okay, so if you have, so here's the R groups, right? And if these R groups are large branched R groups, they're going to sterically inhibit the formation of an alpha helix. Amino acids that can steal hydrogen bonds from the backbone. So if there's lots of hydrogen bonding opportunities in these R groups, they can break the alpha helix. Prolines are too kinked and glycines are too flexible to be involved in an alpha helix. If we measure the propensity of different amino acids to be involved in an alpha helix, what we find is alanine is the most likely to be in an alpha helix. And so everything in this little table here is in relationship to alanine, is relative to alanine. And the higher the number, the least likely you will see that amino acid in an alpha helix. So for example, we talked about proline not being in alpha helixes. Sure enough, it's got a really high value here. Glycine, where's that? Same thing, quite a high value. So make sure that you can read this table. Now, what this unit is, it's really unnecessary to know. Just know that the higher the number, the less likely the amino acids are to form an alpha helix. Beta pleated sheets are the second form that of secondary structure. And what these are when the chain sort of folds back on top of each other, forming this kind of planar sheet where the R groups are sticking out of that sheet. And in alpha helices, those hydrogen bonding that was occurring was 4 amino acids apart. In a beta pleated sheet, the hydrogen bonding that is occurring between the oxygens and the hydrogens off that nitrogen are many, many, many more amino acids apart than just 4. And so we can see here, if you look at a ribbon structure, we can see the blue arrows represent beta sheets. And then up here, this winding ribbon, these are alpha helices. And this last thing right here, these sort of turns, these yellow turns that connect one pleated sheet to the next, or connect a pleated sheet to an alpha helix, is called a beta turn. Now these beta turns take up 1, 2, 3, 4 amino acids, and they make a 180-degree turn. And oftentimes what you'll see in these beta turns are prolines, because they're already kinked. They've got this very kinked nature. And glycines. Glycines, remember, have just a hydrogen as an R group, and so it makes it very easy to take these kind of these 180-degree turns. Finally, we get to the tertiary structure. Now, every protein has primary, secondary, tertiary structure. Only some proteins have quaternary structure, as we'll get to in a second. Now, the interactions essential for the tertiary structure are the R groups of those amino acids. So up till now, we haven't really looked at the R groups except for sort of interrupting the alpha helices. But now we're looking at interactions between R groups of amino acids quite far apart, potentially, and patterns of disulfide bonding. So here we have the different types of weak interactions in the disulfide bridge that contribute to the formation of a tertiary structure. So here we have an R group that has a hydroxyl group. Here we have this R group here, and we've got a hydrogen bond. Here we've got R groups that are all very nonpolar, hydrophobic, and so because of those hydrophobic interactions, they sort of clump together. And then we have, of course, an ionic bond or ionic interactions. And if we see now in a protein, you can see all three levels of protein structure at the same time. And what I mean by that is the primary structure is simply the sequence of amino acids within this chain. The secondary structure are the beta pleated sheets that you see here and the alpha helix that you see here. The turns, the beta turn there, another beta turn here. And then all of those secondary structures fold over on top of each other to accommodate interactions between the R groups of amino acids. And so that's what you're seeing here, everything kind of folding on top of each other. Now, tertiary, they're unlike secondary structure where you have a discrete beta pleated sheets and alpha helices in these folds. You don't have that sort of those categories in the tertiary structure here. Instead, you have more broad categories. Globular proteins are going to be the type of tertiary structure that we're going to see mostly in this class, where everything is sort of quite compact and folded on top of each other. So basically, if you take a look at protein serum albumin, if you were to assume the entire protein was in the beta conformation and didn't go beyond that second level of protein structure, it would look to be about this big. If instead the whole thing was an alpha helix, it would be about that big. But instead, you get this globular nature, this third level of protein structure, the tertiary structure. You form a globular protein that is this relative size right here. So globular proteins are quite compact. What you can start to do, though, when you compare lots of proteins to each other and kind of pull out these patterns, what you see are motifs. These are a group of folds that you see over and over and over again. Things like the beta alpha beta loop or the beta barrel. Okay? They may or may not be independently stable. So if you rip them out of a protein, they may not be able to fold correctly or in a functional thing without the rest of the amino acids around them. That is more of a domain. So domain, again, it's a portion of a protein that has a very specific set of folds. Unlike the motifs, these oftentimes are have distinct function and they are independently stable. So, for example, DNA binding domains, they allow the protein to bind DNA. Activation domains, they allow that protein to interact with other things. Activating domains, catalytic domains, these are parts of a protein that catalyze reactions. And because these are domains, you can cut this off, it'll fold properly and still maintain that activity. And so you could transfer them one domain to another protein to another protein. And you can start to engineer proteins that way, and that's going to be an important part of the journal article you're reading. Finally, quaternary structure only occurs for proteins that are made up of multiple polypeptides, multiple subunits. So if it's made up of 2 polypeptide chains, we call it a dimer. If it's made up of 3, we call it a trimer. If it's made up of 4, a tetramer. And if it's a bunch, sort of undefined, then we call them oligomers. So for example, here we have hemoglobin, and it is made up of 2 alpha chains and 2 beta chains. So it is a tetramer. And the way in which these 4 different chains come together are those same weak interactions. They are not necessarily covalently bound to one another, although they can via disulfide bridges, but it can be where they're just loosely associated with each other via these weak interactions. So make sure that you go back and you review this lecture, and you are able to explain the importance of the Anfinsen experiment, that you can describe the characteristics of the primary structure of proteins that limit the way that chain is going to fold. Describe the primary interactions that are necessary for the second, third, and fourth levels of protein structure. Differentiate between alpha helices, beta pleated sheets, beta turns. And finally, explain the difference between protein motifs and protein domains.